Hello, one and all. The Chapter 2 promised video lecture, Chemical Foundations of Life. So I kind of view this, we'll start out by saying that I kind of view this as a review of chemistry classes you might have had in high school, biology classes you might have had in high school, the same for college level classes, a little bit in an anatomy class at the beginning of Anatomy 101, this was covered. And so I kind of feel like a lot of this was covered, but we're going to up the ante by adding some connections to physiology as we go through this. So that's the kind of stuff that you want to look for as we progress through the topics. So our objectives today are to describe the atom and then how that's connected to the human body. Molecules, bonds, how that's connected to the human body. We're going to talk about solutions, which we did in the lab. We want to know the different parts of solutions, so solvent and solute, and their importance in a solution, their role in a solution. We're going to talk a little bit about pH and how it affects proteins in our body, which is fundamental to health. We're going to compare and contrast the four biomolecules. And remember, those are carbohydrates, lipids, proteins, and nucleic acids. So to start out with, an atom is the smallest unit of an element. And I remember in chemistry class being confused at the difference between an atom and an element. And the best way that I can explain that is that remember how we talk about when we set up graphs in the lab, we always have a variable, and then we have the unit that that variable is measured in. For example, mass is measured in grams, mass being the variable, grams being the unit. So the way I think about this is that an atom is a unit of an element. So in the same way that grams are a unit of mass. An atom is composed of protons, neutrons, and electrons. Two of those are charged particles, one of them is not. In terms of size, protons and neutrons are 10,000 times bigger than electrons, so they tend to give the mass to the atom itself. Depending on what element it is, that's going to determine what the mass is, because each element has a different number of protons and a different number of neutrons. So the more protons and neutrons you have, the bigger the mass of the atom. Electrons then, because they're 10,000 times smaller, are not really going to affect the overall mass. What they do affect is the Brownian motion that we talked about in our lab, in our diffusion lab. So electrons are traveling at about 1,000,000 miles per hour. You have the same number of protons as you do electrons. So if you increase the number of electrons, then you've got more objects moving at 1,000,000 miles an hour and therefore more Brownian motion. So the number of protons, neutrons, and electrons that an atom has determines a couple of different things. It's going to determine the charge of the atom. Most atoms are neutral in charge because they have the same number of protons as they do electrons, so the opposite charges cancel each other out mathematically. The size, as I said, is going to be determined by how many protons and neutrons an atom has. And then most importantly, in terms of interaction between atoms, the number of electrons in the outer shell, also known as the valence electrons, are going to determine how an atom bonds to another atom. So here's a visual of atomic structure, just so you get an idea of how these subatomic particles are related to one another. If you remember, protons and neutrons are found in the center of an atom. How do we know this when we've never even looked with the naked eye at an atom? We know this from over 200 years of indirect evidence, kind of being compounded to help us to believe and make decisions based on this model, that protons and neutrons 10,000 times bigger than the electrons are compacted into the center of an atom. kind of looks like a solar system. That's what I remember the first time I saw it, is it looks like a sun with individual planets traveling around it in orbits. And in fact, we call the space around the nucleus electron orbitals. And that's where the electrons travel as fast as they travel. Because they travel this way, the orbital that is closest to the nucleus holds the fewest amount of electrons. In fact, it can only hold 2 electrons because they're traveling so fast. The analogy that I like to use is if we took the speedway in Indianapolis and we cut it down to an eighth of the size, but we allowed the same number of cars to travel around that oval, they would crash into each other because there's just not enough space for them to travel at that speed without doing that. It's the same type of thing. This orbital happens to be the closest to the nucleus and the smallest, and because of that, only 2 electrons can travel in it. As you start to move away from the nucleus, as these orbits start to move away from the nucleus, the second orbital, the third orbital, they can hold up to 8 electrons or more as you move further away. The outermost orbital of electrons, the one that is furthest away, whether it is the first one as it is in hydrogen and helium, the second one as it is in some of the larger lithium, etc. elements, or the third one because you have enough electrons that they can't all fit in the first and the second. Those orbitals, then the very last orbital is called your valence orbital, and it contains your valence electrons. And those are the only electrons that are involved in bonding. So when you look at the periodic table, which is the way that we organize the elements so that we can have some understanding of them, this is a physiology version of it. And although all of the main elements are listed on this periodic table, what we're focusing on is the elements that are essential for humans, those that are in purple, the ones that are suggested as essential for humans, and then the ones that are non-essential. Typically, the non-essentials are not involved in the human body either because they are toxic, or if they're in this list right here, they're incapable of bonding. So they cannot make molecules that are beneficial for our life. The fundamental elements of living molecules are carbon and hydrogen. Those are the 2 elements that are involved in all organic substances, substances that are found in life. When you add nitrogen, oxygen, and phosphorus to that carbon and hydrogen, you get combinations of molecules called macromolecules. And those are our 4 molecules that we focus on when we are trying to understand what compounds are essential for both the structure and the function of a living human being. So we're talking about combining carbon, hydrogen, and oxygen in carbohydrates and lipids, and carbon, hydrogen, nitrogen, and oxygen in proteins, and then you add phosphorus and you get your nucleic acids. All of these other support elements are elements that are essential for structure and supporting structure in our body, so anatomy. And then these 2 right here in particular should be something that you recognize immediately as associated with an action potential. So these are associated with the nervous system and being supportive in electrical conduction along an axon in a neuron. Iron, for example, an ion that is attached to hemoglobin. Very, very essential for carrying oxygen and carbon dioxide throughout the bloodstream for use by the cells, and then for expulsion of carbon dioxide out of the body. And all of these others have very specific jobs as well. Magnesium is one that's in social media a lot because magnesium citrate and magnesium calcitrate are supposedly health and wellness type supplements that will help you with constipation in the case of MagSit, and calcitrate helps you with I believe it's muscle Charlie horses and things like that that need magnesium as a support element. So we're going to talk about what is the role of the atom in the body. And the first thing is that it forms molecules and macromolecules. So our first example of a building block of a macromolecule is glucose. Glucose is a monosaccharide. It's a sugar. It has a cyclical structure to it with some things hanging off of it. And we don't need to be worried about those right now. But glucose is a macro energy source for our body. So how do atoms play a role in that? Well, each of these atoms are bonded to one another, and in those bonds, energy is stored. So every time your body breaks one of those bonds, it's going to release large amounts of energy. And then the mitochondria in your cells are going to take that large amount of energy and package it into smaller packages of energy called ATP. And that's over here on the right side. So these are micro energy sources. So imagine that you have these building blocks inside of the mitochondria. You've got these three building blocks inside of a mitochondria. You bring some glucose into the cell, you break some bonds, and you take the energy that is released to be able to attach these phosphate groups to the backbone of ATP, adenosine. So this is called adenosine. Try 1, 2, 3 phosphates, and the energy from glucose is what is trapped inside of these smaller units. And then the cell can use this unit as necessary in a much more efficient manner. Another job of molecules and macromolecules is lubrication and bonding. So water, because it is a polar substance, it has a charge. So this side has one charge, this side has another charge based on unequal sharing of electrons by covalent bonds. It enables water, this hydrogen, to attach to other molecules, and it enables the oxygen to attach to other molecules. And so it provides bonding between one molecule and another molecule to either move it somewhere, to change its structure, to degrade it. But it also plays a role in lubrication for moving things throughout the body. Another important part or job or function of atoms in the body is that they enable electrical activity via ions. So when an ionically bonded compound like potassium iodide, which has one potassium ion with one valence electron attached to one iodine atom that needs one electron to have its outer shell filled with electrons. When that happens, so if you put some calcium and some iodine close together, those iodines are going to align themselves with individual potassium, sorry, I know I was calling it calcium there for a minute, with individual potassiums so that their outer shells can be happy. So if you remove an electron from the valence orbit of potassium, the next shell down is happy. it's got a complete number. This little electron hanging out there by itself in the outer shell is not happy because that outer shell isn't filled. So if it just gives up its electron to iodine, whose outer shell only needs one electron, then both of them are happy. And as a result, they're ionically bonded. Now, in our body, the benefit of that is if we are to take in potassium iodide, we can then break it down and get individual potassium ions and individual iodine ions. Iodine is responsible for correct thyroid function. So your thyroid as an organ in your neck needs iodine to function correctly, and it needs it in the form of an ion. So when we have ionic compounds that we're ingesting or we're making in our body, we can then break them down as necessary to get the individual ions that we need. And again, let's back it up and think about how potassium is involved in an action potential that creates electrical activity on the neurons. So here's what I'm talking about. And again, I believe this is review. We're going to work on this a little bit because I think it is a confusing topic, but I believe in Anatomy 102, this was a topic that you talked about during the nervous system unit. Maybe it was in 101, I'm not sure. What we're looking at here is if we go from left to right, the orange part is a neuron's axon. So that long stem that comes out of the cell body of a neuron is represented in this orange area. Okay, outside of the orange area is the extracellular environment that we talked about, that is a solution compartment in the body. And remember, I'm just connecting this back. Extracellular environment has less water than the intracellular environment has. Now, water is not one of the molecules that we're worried about in creating an action potential, but I just wanted to kind of get you oriented as to where we're at. So we have taken a neuron and we're focusing in on just a small portion of that long stem that comes off of the cell body. Normally, the neuron is going to be inside of the cell. The cytoplasm is going to be negatively charged. The reason that it's going to be negatively charged is because it has more negatively charged ions, not shown here, but more negatively charged ions in this space than outside of the cell. The reverse is true outside of the cell, and that's where these pluses come from. In the extracellular environment, there is more positively charged ions that give an overall positive charge to this space. To be able to reach threshold and cause electrical activity to travel down this neuron axon, we need to flip the charge. So if you look here in this first box, outside is positive, inside is negative. The way that we're going to switch or flip the charges is we're going to bring sodium ions through protein channels into the axon. Okay, so now we have a lot of potassium positively charged ions, and we have a lot of sodium positively charged ions. In comparison to the extracellular environment, this is now more positively charged than this space. And so we are able to flip the charge. And by doing so, we have done what is called depolarization. We have started electrical transmission down the axon so that a signal can be sent from one end of the neuron to the other end of the neuron, to the synaptic cleft, if you remember that. Well, we don't want the electrical activity to stay here. We want to propagate it down the axon. So now we have to flip the charges again. To repolarize the area, we're going to move potassium out of the cell. So we're taking away positivity from the positive environment and therefore making it more negative. So we remove potassium this time to the outside environment. It becomes more positive, and this is left with a negative charge, just like it was in the beginning. That is called repolarization. And the importance of this is this enables us to meet threshold again. So if another signal comes along, we can depolarize. If we remain in this, we can't repolarize, and that signal will be stopped. Okay, and sometimes one axon is going to have to propagate multiple signals. So to speed the process up of making this, so the flip over from negative to positive, making it completely positive outside again and completely negative inside again as it was before depolarization, we use what's called a sodium-potassium adipase pump. And for every 3 sodiums that we move back to the outside environment, we move 2 potassiums to the inside environment until we can quickly get it back to pre-depolarization state. So this is kind of an intermediate state. This is what's going to get us back to this state where we're ready to depolarize again and send another signal. So again, just a perfect explanation of the importance of atoms in the human body. We will get into this much later in the course when we're studying the urinary system. We actually do a lab on this. But one of the third things and most important things that atoms do for the body is they help to maintain an acid-base balance. In other words, they're helping to maintain the pH of the solutions that are found in the body. So we have extracellular solutions, we have intracellular solutions, we have blood plasma, and the example they give here is the narrow range of pH that must be maintained in the blood for the blood to do its job, which is transportation of gases, transportation of hormones, transportation of white blood cells for immune processes, transportation of water, transportation of glucose and other nutrients from our food. So it's super important that we keep the pH in a proper range in each of those solution compartments so that the metabolic chemical processes that have to be done in each of those environments can be done appropriately. The most important thing to know about pH is that pH supports proper protein structure. If you go too acidic or too basic in any one of those compartments, any of the proteins that are in those compartments can denature. And proteins act as enzymes. They act as structural components on the cells for cell signaling. They are hormones. And if you denature any one of those, they can no longer properly function in the chemical equation or reaction that they're trying to help with. So it's critical that we have chemicals in our body made up of atoms that can buffer those solutions so that we maintain narrow ranges of pHs and we don't let those substances become too acidic or too basic. Another function of atoms is the fact that an atom can have a single type of atom can have a different number of neutrons. For example, carbon can have an atomic mass of 11, an atomic mass of 12 or an atomic mass of 13 or 14. And if you look at the periodic table, the atomic mass of carbon is listed at 12 point, I believe, 001. That's kind of weird because if the atomic mass reflects how many protons and neutrons the nucleus has, it seems like it would be a whole number, right? Because the atomic number of 6 tells us there's 6 proteins. So shouldn't the mass number be 12? So instead of 12.001 to tell us that there are also 6 neutrons, because 6 plus 6 equals 12. It doesn't equal 12.001. Here's the problem. There are many different types of carbon. There is carbon with 6 neutrons. There's carbon with 7 neutrons. There's carbon with 8 neutrons in it. And there's carbon with 9 neutrons in it. If we listed each of those separately on the periodic table, our periodic table would take up a 6 by 6 foot piece of paper. And so what we've done instead is we've taken all of the isotopes of one element and we have added up their individual masses, divided them by the number of isotopes they have, and we come up with an average atomic mass. And that's what we list on the periodic table. But what we know is that carbon-14, for an example, a specific isotope of carbon, is found in the bones of living things, including humans. It is a radioactive substance because it has a different number of protons than it does neutrons. So it think of it as making the nucleus a little bit imbalanced. And because of that imbalance, there's a chance that the nucleus is going to break apart. And it actually does. And when it breaks apart, it gives off particles, radioactive particles, and it gives off light, which is a form of energy. And so most isotopes are associated with radiation. And so when we say that something is giving off radiation, it is the atom's nucleus that is giving off light and particles because its nucleus has a different number of protons than it does neutrons. And the bigger the difference, the bigger the amount of radiation in general. So we figured out how to use that to our benefit. And we know that if we can capture, because it gives off that light, if we can get some of an isotope embedded in a cancerous tumor, for example, and we can take a picture of that by using a scanner, we can see a cancerous tumor glow. So if there is a substance in a cancerous tumor that will absorb the glowing isotope, then we can capture a picture of that and we can show it so that we know that there is a cancerous tumor in this brain scan, for example. So this is called a PET scan. So the benefit of this is that we can find and learn how to surgically remove or treat a cancerous tumor. And it can be used for finding other masses as well and determining what kind of mass it is when it's an internal mass that we cannot see by palpation or that we can't take off of the skin, for example. The benefit of that is that we can do that. The downside is, and why we don't do this a lot, why we don't give PET scans a lot, is first of all, the equipment is very, very expensive, but also because you have to get that radioactive isotope into the body some way. And it's typically done through an IV, and large amounts of any radioactive substance are not going to be safe for a patient. So we want to do it sparingly. We want to do it only when it's necessary. And so that's why you don't see a lot of this. But our knowledge and understanding of isotopes have led us to be able to do this. We also use isotopes in the treatment of cancer. So that thing which is negative about isotopes becomes a positive when you target it during radiation therapy at a cancerous cell mass. You can now blow up that cancerous cell mass with this radiation coming out of the nucleus of the atoms involved, and use it for a positive reason. So stepping it up, when we take two atoms and we bond them together, we get a molecule. Now, there's a little bit of weirdness going on here. In our book, our text uses the word molecules for both molecules and compounds. Okay, so molecules typically describe when you take one type of atom and you bond it to the same type of atom. For example, O2. That's two oxygens bonded together. Compounds are typically when we take two different elements like sodium chloride, sodium and chlorine, or even water. Think of water, hydrogen and oxygen, two different atoms, and we bond them together. So I know that I have not heard anybody saying, I have a compound unit of sodium chloride. We always say there's a molecule of sodium chloride. There's a molecule of oxygen gas. There's a molecule of water. So this is kind of a technicality. We're going to go with what our text says, and that is that anytime we're describing a unit of a compound, we're going to call it a molecule. Anytime we're describing a unit of an element, we're going to call it an atom. So there are three types of bonds in review. Covalent bonds are the strongest bond. That is a test question, by the way. Ionic bonds provide us with electrolytes, as I explained before. And hydrogen bonds are involved in a couple of different things. The properties of water, which is the solvent in our body, the primary solvent in our body. They are responsible for protein structure, and there are 4 different levels of protein structure. And they are responsible for the DNA double helix. So they hold those nitrogen bases together until a gene needs to be opened and copied into a protein. So looking at a visual of them, here's our ionic bond. This electron in the valence shell is completely given up to another type of atom, and as a result, they bond. In covalent bonding, you are sharing electrons between two different elements. In hydrogen bonding, you are sharing electrons between two different molecules. Do you see the difference here? This is two different atoms that are sharing electrons. This is a molecule of water. This is a molecule of water. And this is the bond between the two different molecules. So covalent bonds can be polar or nonpolar. Polarity is determined by whether the electrons are being shared equally or unequally. On the left, you see water. On the right, you see methane. Water is an example of a polar covalent bonded molecule because the oxygen side actually has a different charge, negative, than the hydrogen sides, because you have two sides, hydrogen here and hydrogen here, which are positively charged. And it has to do with the fact that these electrons are traveling a million miles an hour, and sometimes all of them are going to be found in one place and only some of them at another time. And so it's an uneven sharing. Whereas in the methane example, those electrons are only shared in one spot. So this hydrogen needs 2 electrons to be happy. This carbon needs 4 electrons to be happy. So when these travel around, they're only traveling around that one atom. And so it's much more stable, and therefore you do not have charges on any of these hydrogens or the carbon itself. Even though they're both sharing electrons, they're doing it in a different way. The polarity of water is what makes water behave the way it does. So water likes to bond to water. I told you that because you have a negative charge on the oxygen of one water, you have a positive charge on one of the hydrogens of another water. Opposites attract, so they bond together. In the same way, water is attracted to ionic compounds. So if you put salt in water, water is going to be attracted, both its negative oxygen part and its positive hydrogen parts, to the different sides of each of those ions, and it will actually pull those ions apart. Well, that's a good thing for us because remember, we need sodium, we need chlorine, but we don't need sodium chloride. And so if we have um, salt brought into our body and it becomes a part of one of the solution components in our body, water is going to work to break it apart so that we're able to get both the ions that we need to perform in other chemical processes. Again, ionic bonds are giving up electrons and gaining electrons. Hydrogen bonds are responsible, so they're going to occur right here in between these nitrogen bases that hold together the two sides of the double helix. Remember A to T and G to C. In chapter 3, we're going to hit that pretty hard again, protein synthesis. But for now, what we need to understand is that a hydrogen bond exists between these to hold the double helix together. Hydrogen bonds exist between amino acids to be able to create the 4 levels, 1, 2, 3, and 4 levels of protein structure. Okay, and again, we'll look at that a little bit more in depth later. They are the weakest bonds. Covalent are the strongest, hydrogen are the weakest. So what are the importance of bonds in summary? They store energy for cellular work. We talked about that with glucose giving up its energy to ATP. They package materials for movement, for tight spaces, and for specific shapes, protein, for example. They regulate pH and temperature of blood plasma, necessary for all metabolic function. They recycle atomic material and energy in the body so that collectively all of these things are involved in our metabolism. And by definition, metabolism is all of the chemical processes and reactions that are going on in our body. When we did our osmosis lab, we dove into the idea of what is a solvent and what is a solute in relation to a solution. So blood plasma, for example, is a solution. It has many, 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 many solid components. Those are solutes. All of these are dissolved in water, which is the liquid portion of the solution. So blood plasma is a combination of water and many, 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 many different types of solutes, all listed in different colors here. And if you start looking through some of these, you're going to recognize some of them as being involved in immunity and wound help. These things are going to break down pathogens that come into your body, and then a lot of things that are involved in movement in terms of moving things from one place to another. How do we use in like what's the clinical relevance of solutions in general? Complete blood panels we've learned, and urinalysis and IV fluids. Those are the three things that you're just going to get beat over the head with as a practitioner. Either you're responsible for gathering it if you're a phlebotomist in these cases, or you're responsible for giving and pushing IV fluids or IV medications if you work in a rheumatoid or a cancer unit. In any case, we're talking about either body's solutions and how they show us our health or us being unwell, and then how to use external solutions to bring those back into balance and or health. I'm going to leave this as it is. It is a video that you need to watch, and some people have said that they've had a hard time getting it to play. First thing I want you to try is to put the PowerPoint in presentation mode and then press play, and you should be able to play it. If you can't, look at the title up here and type it into YouTube, and you should be able to pull up the same video. One of the things that's going to be talked about in that pH video is enzymes and their role in chemical processes in the body and their role as it's related to pH. And so you first have to understand that an enzyme is a protein and it has a particular shape. That shape is directly related to the substance that it's going to attach to. After all, an enzyme is designed to speed up a reaction. We have so many reactions in our body that have to occur at lightning speed to be able to provide a response to either external or internal changes. And as a result, we have to have what are called catalysts, and that's what enzymes are. And they bind a substrate, another chemical, that is the mirror image of their shape, so that that chemical reaction can be speeded up. If it loses its shape, let's say this ridge, and this ridge are denatured so that it's completely flat here. And the way that an enzyme gets denatured is that the pH of the substance that it's dissolved in, so blood plasma, for example, is too high or too low for the hydrogen bonds to be able to keep this protein in its shape, in its appropriate shape. And so if we lose those three ridges there, this is not going to be able to fit into its substrate, and the chemical process that it's responsible for will not happen. Now you can imagine if this only happens to one enzyme, no big deal. If it happens over a long period of time because your pH is unbalanced and not being buffered, now we're talking about multiple chemical reactions not being able to happen. And then you're going to start seeing signs and symptoms of the body being unwell. Two examples of enzyme disorders. And when I spoke about this in class the other day, a phlebotomist who has done multiple urinalysis tests said that she has seen the maple syrup urine of a person with this disorder that lacks an enzyme that breaks down proteins or amino acids from the blood, and that it literally looks like maple syrup. So this is an example of a person that genetically has lost the ability to create the protein that makes the enzyme that is necessary to break down amino acids. So as a result, those amino acids are being passed out of the body in the urine, and it's changing the urine's consistency and making it very thick. Tay-Sachs disease is another enzyme-related disease. Babies that are born with Tay-Sachs lack a particular enzyme that breaks down fats in the central nervous system. And you can imagine the negativity of having fats building up on the central nervous system, because fats behave as insulators in electronics. And to have insulation around your brain, which is an electrical factory, essentially, we don't want insulation. So babies with Tay-Sachs disease typically die within the first weeks of life because of this lack of an enzyme that is related to the process of breaking down fats in the central nervous system. So here are our 4 biomolecules. We finally come to this, and flat out memorize this chart. Carbohydrates are made up of monosaccharides. That means it's their smallest part, their monomer. If you put a bunch of monosaccharides together, you get a polysaccharide. An example of the simplest monosaccharide is glucose. Lipids, they're made up of fatty acids and glycerol. 3 fatty acids to every 1 glycerol. Okay? That's called a triglyceride. Another example of a lipid is a phospholipid, and that's what makes up the cell membrane and the organelle membrane. Now, a phospholipid only has 2 fatty acids and a phosphorus head, but it's another example of a lipid. Okay, so these are your base units for a triglyceride. Another example besides a triglyceride of a lipid is a phospholipid. And a phospholipid is only slightly different in its makeup in that it has 2 fatty acids and a phosphorus head. Proteins are built from 60 different amino acids. An example of a protein is an enzyme. Nucleic acids are built up of nucleotides. I should have put nucleotides here, so that's a mistake. Nucleotides have 3 parts to them: a phosphate, a sugar, and a nitrogen base. Now, if you're DNA, you have deoxyribose as your sugar. If you're RNA, you have ribose as your sugar. If you're DNA, you have A, T, G, and C as your nitrogen bases. If you're RNA, you have A, U, G, and C for your nitrogen bases. A third example of a nucleic acid, because of its building blocks, is ATP. What's the role of a carbohydrate? It's the primary energy source of our body, and it also plays a structural role. Here's a picture of glucose, the monomer of most and many carbohydrates. Notice its cyclical appearance and that it only has 3 types of atoms: carbon, oxygen, and hydrogen. If we want to make a carbohydrate, we have to use a process called dehydration synthesis. This process is where we take 2 monomers, for example, a glucose and a glucose. We remove a hydroxide, OH, from one of them and a hydrogen from another. That makes water, right? 2 hydrogens and an oxygen makes water. So you remove a water and you bond together the 2 glucoses. You now have, instead of a monosaccharide, you now have a disaccharide. If you want to add another glucose to the end of this, you do the same process. Take an OH off of this, take an H off of the next monosaccharide, remove that molecule of water, and now you have a trisaccharide, and you can keep adding them as you go. If you want to break down a biomolecule, such as a disaccharide, you add that water back to it, and it will break that bond so that you have 2 individual monosaccharides. That process is called hydrolysis. So you're going to hear about these 2 processes over and over and over again. The making and breaking down of biomolecules in the body. The purpose of a lipid is for energy storage and to make membranes, but steroids are also an example of lipids. So triglycerides are for storage, phospholipids are for membrane structure, and steroids are used for cell signaling. They're usually in the form of hormones, and we know that hormones are involved in the immune response. They help to establish bone health, and they help to keep our cell membranes from falling apart. So 3 different types of lipids: triglycerides, phospholipids, and steroids, all with 3 different jobs. Sorry about my cat. He doesn't talk much, but of course, when I want to do this, he's got something to say. There's a picture. This is a triglyceride, huge compared to our glucose. This is our phospholipid, only 2 fatty acids. This one has 1, 2, 3 fatty acids. This one has 2 and a phosphorus head. And then here's our steroid. And a steroid is typically 1, 2, 3, 4 cyclical carbon rings stuck together. Proteins, as we said before, are made up of amino acids. Here is their function: enzymes to speed up reactions, cell receptors for cell communication, and then transporters to move things in and out of the cell. Their structure or shape determines their function. So shape is extremely important when we're talking about proteins. So here's an example of an enzyme and how it's folded. And these would be the functional areas where the substrates could attach to the rest of the protein. And when those 2 substrates actually attach to the protein, they change the entire shape of the protein, causing the chemical response to move on. This is a picture, if we go back to the last slide and we talk about cell receptors, these are for cell signaling, for saying, hey, I'm this type of a cell attached to me, and I will help you to tell this cell what to do. Please watch this video, which goes in depth into protein folding, the 4 different types of folding, the importance of the shape modifications, etc. Again, if you have trouble pulling it up, try putting it in presentation mode first, and then if that doesn't work, use this title and put it into YouTube. And finally, we look at a comparison between DNA and RNA. And what I want to remind you of here, all we're going to do here for this chapter is ask that you understand the difference between DNA and RNA. And there are a couple of differences. First of all, DNA is only found in the nucleus. RNA can leave the nucleus. Secondly, DNA is made of 2 strands. RNA is made of 1 strand. DNA sugar is deoxyribose and RNA is ribose. And then there's that one difference between nitrogen bases between the 2 of them. So if you ask why can't DNA just open up and make a protein itself, DNA has to stay in the nucleus to be protected. DNA is a book of all the proteins that can be made in a human body. If we let it exist outside of the nucleus, bacteria, protists, any kind of pathogen that gets into a cell could attack it and break it down, and we would lose all of our directions for making proteins. On the other hand, if we make a copy of only 1 gene that is capable of leaving the nucleus, if it gets attacked by a pathogen, no big deal, because we still have the book of directions left over to be able to make another mRNA to make that protein. So it's a protective measure to protect this vast amount of information. DNA provides the code for making the proteins. It's divided into genes. And in chapter 3, we'll talk about transcription, and then we will talk about translation in making the protein. So, how do you get a 4-meter piece of DNA to fit inside of a cell that we have to blow up 400 times, magnify 400 times to be able to see under a microscope? It's a lot of folding. That's what I'm going to tell you, a lot of folding. And when you stretch it out, you get this double helix. When you open up the double helix right here for an individual gene that codes, let me see if I can go back. Of course I can't, but an individual gene that codes for a protein, you're going to get that protein. You put those proteins together, maybe 3 or 4 of them, and you get, for example, hemoglobin, which is found on the surface of your red blood cell. Hemoglobin is made of multiple protein compounds that are bonded together to do a particular job, and that is to carry oxygen and carbon dioxide throughout your body. So don't worry yet about transcription and translation. We will go into detail in that in chapter 3. So that ends the lecture. Please be sure to go back and look at those 2 videos and then complete the lecture activity that is in module number 2. If you'd like for me to check that module activity, I will definitely do that. You just send a picture of it and I'll tell you whether you're on the right track or not, or you can bring it to class with you on Tuesday. Sorry for the delay in getting this posted. Have a great weekend. Stay warm, stay safe, and hopefully I'll see you on Monday.